Let's talk about diffuse large B-cell lymphoma, or DLBCL. It's the most common aggressive lymphoma out there. And while a lot of people can be treated successfully, a huge chunk, we're talking 30 to 40% of patients either don't respond to the standard therapy or they relapse. For decades, this has been a massive puzzle for doctors. And the reason why? Well, it turns out DLBCL isn't just one disease. It's actually a whole spectrum of different diseases all hiding under one name.
Okay, so this right here, this is the question that's been driving decades of research. It really gets to the heart of the whole problem. You can have two people walk into a clinic, and under the microscope, it looks like they have the exact same cancer. So why does one person respond beautifully to treatment while the other person's cancer just doesn't budge? The answer, it turns out, is hidden deep inside the tumor's own biology.
So in this explainer, we're going to peel back the layers of this really complex disease. We'll start with that initial puzzle. Then we'll look at how scientists first started splitting the spectrum using something called gene expression. After that, we'll cut even deeper with genetics. We'll see how all this new knowledge is changing things in the clinic, how the cancer itself evolves to fight back against treatment, and finally, what's on the horizon, the next frontier.
Right, so for a huge number of patients, the standard treatment, a chemotherapy regimen called RAJA-PATHOLOgy, just isn't cutting it. And that clinical reality, that was the big red flag. It told scientists that something much deeper was going on, that just lumping all these cancers together as DLBCL was covering up some absolutely crucial biological differences. They knew they had to find a better way to classify this thing.
For the longest time, this is pretty much how we saw DLBCL. Just this single, messy, complicated disease. You can see on the left here, this jumble of different-looking cells all gets funneled into one single category. It's kind of like looking at white light. You know it's light, but you can't see all the individual colors that are actually making it up. The big challenge was finding a tool that could act like a prism and split that light apart.
And that first tool, that first biological prism, was something called gene expression profiling, or GEP. Instead of just looking at the cancer cell's shape, researchers started measuring which genes were actively turned on or off inside them. This gave every single tumor a unique molecular fingerprint. And honestly, it was a total game changer. This visual here does a fantastic job of showing what happened. GEP acted like that prism we were talking about. The single beam of light, representing all of DLBCL, went in, and what came out were these distinct bands of color. These were the first major subtypes, and they were named after the normal, healthy B cell they looked most like. They called them the cell of origin subtypes. So the two main subtypes that popped out were the germinal center B cell-like, or GCB, and the activated B cell-like, or ABC subtype. And this is the really crucial part. This wasn't just some academic exercise. The data clearly showed that patients with the ABC subtype had way worse outcomes on standard therapy than patients with the GCB subtype. For the very first time, we had a real biological reason for why patients were having such different journeys.
But, gene expression was only the first split of the light. It told us what the cancer cells were doing, but it didn't really tell us why they were doing it. To get to the root cause, scientists had to go a level deeper. They needed to read the tumor's fundamental instruction book, its DNA, so they started sequencing the genetic code of these cancers.
Let's go back to our prism analogy, because this visual adds the next chapter. If GEP was the first prism, you can think of genetic sequencing as a second, even more powerful prism. It takes those first colored bands that came out of the GEP prism and splits them again, resolving them into even more detail. You can see on the far right, we now have this much more granular, much more precise set of categories. These are genetically defined groups that give us a much sharper picture of the disease.
Now, I know this looks really complex at first glance, but it's an amazing way to see what defines these new genetic groups. Each of these colored clouds is a different subtype, and the words inside, those are the genes that are most commonly mutated in that group. So, for example, take a look at the blue cloud on the bottom, called MCD. It's defined by mutations in genes like MYD88 and CD79B. And then you have the dark red group, EZB, which is characterized by things like BCL2 rearrangements and mutations in KMT2D and CREBP.
And this is a really key takeaway. What's so wild is that different research groups around the world were developing their own genetic classification systems, but they all kept landing on the same core patterns. And that was huge. It confirmed that these weren't just random collections of mutations. These were real, consistent biological diseases with shared foundations.
So why do we care about all this? Well, this little plot on the bottom left tells the whole story. It shows patient survival, but it's broken down by their genetic subtype. And you can see it plain as day.
Some of these groups, like the red MCD line and the purple N1 line, are linked with very poor outcomes. But others, like the light blue SG2 line, tend to do much better. This directly connects a tumor's genetic code to a patient's prognosis, and that brings all this molecular science straight into the real world, into the clinic.
Okay, so understanding the code is one thing, but using it to actually help patients, that's the ultimate goal. This new, incredibly detailed understanding of DLBCL biology has kicked off a whole new era of precision medicine, where treatments are being designed not just for DLBCL in general, but for its specific, individual biological subtypes.
This table gives you a great snapshot of this new reality. Researchers are running trials that go after the specific weaknesses of each subtype. But it's definitely a story of trial and error. Look at the Robust trial. It targeted the ABC subtype, but, well, it didn't show a benefit. But then look at Remodel B. They added a different drug, bortezomib, and bam, a significant survival benefit for that exact same ABC group. And just recently, the PolarX trial showed a powerful new drug combo works wonders, but when they dug into the data, they saw the benefit was almost entirely driven by patients with, you guessed it, ABC-DLBCL. We're finally learning how to match the right drug to the right biology.
But what happens when treatment doesn't work and the cancer comes back? To really beat this disease, we have to understand how it evolves to resist our therapies, because the biology of a tumor that's relapsed isn't always the same as the one the patient started out with.
Okay, let's break this slide down, because it shows two totally different ways that relapse can happen. Look at the diagram on the left first. This is showing an early relapse. See how the original tumor and the relapsed tumor are pretty much identical? That suggests the cancer was probably resistant to chemo from day one. The standard treatment just couldn't kill it. Now look at the right panel. This is late relapse, and it's a completely different story. Here, the original and relapsed tumors have both evolved separately from a common ancestor, something they call a common precursor cell. They went down different evolutionary roads.
So what's the big takeaway here? Well, this actually explains something doctors have seen for years. Patients who relapse late often respond much better to a second round of therapy. And why is that? Because the tumor that came back evolved along a new path. The original tumor might have been wiped out by that first round of chemo, but a new one grew from that common ancestor. And this new one might not have the same resistance tricks up its sleeve, making it vulnerable to treatment all over again.
So, we've gone on this incredible journey from seeing DLBCL as just one disease to a whole spectrum of many different ones, all defined by their gene expression and their genetics. And we've seen how that is already changing treatment. So what's next? What's on the horizon? So where are we headed? Well, the future is all about making these tools even more powerful and putting them all together. That means things like creating one single unified genetic classification system so that every researcher and doctor around the world is speaking the same language. It also means looking beyond just the cancer cell itself and studying its entire ecosystem, what we call the tumor microenvironment. And we're now using incredible new technologies that can analyze the biology of every single individual cell. And the goal of all of this? To fuel even smarter, more targeted clinical trials for patients.
You know, we started this whole journey by looking at DLBCL as a single, confusing puzzle. But by using these increasingly powerful biological lenses, we started to resolve it into distinct, understandable, and clinically important diseases. It's just a powerful reminder that inside all that biological complexity, there's a hidden logic. So the question now is, after all these decades of work, if a cancer's biology is its story, are we finally learning how to read it?